Mosses and liverworts are found in woodlands everywhere. The moss plant consists of crowded upright shoots bearing simple spirally arranged leaves. This is the gametophyte or sex cell producing generation and culminates in the production of sperms and ova. The embryo developing as a result of the fusion of sperm and ovum becomes the sporogonium, which represents the sporophyte generation. This produces not sex cells but spores, which give rise to a new gametophyte generation again. Liverworts are bryophytes, which usually live in damper places than do most mosses, such as river banks, where the air is moist. Marchantia is a thalloid liverwort. It often bears vegetative reproductive organs or gemma cups on its upper surface. Within these, green plates of cells or gemmae are produced. They are washed out by rain. They germinate in suitably damp conditions, producing firstly rhizoids and eventually becoming independent liverwort plants. Frolenia is a liverwort which grows on trees. Its leaves have lobes that hold water. Funaria hygrometrica is often found in bare places where there have been fires. Its life history is typical of that of a moss. Buds on the shoots bear sex organs. The male buds are terminal, the female lateral. The male bud consists of overlapping leaves. A longitudinal section through the male bud shows the club-shaped male organs or antheridia, which are intermixed with sterile hairs. They produce male cells or sperms. Unlike the male, the female buds end side shoots which often produce their own rhizoids. In longitudinal section, the long-necked archegonia are seen to be interspersed with paraphyses. At the base of each long neck is a swollen venter containing a single ovum or egg. The neck canal cells presently break down. The sex cells of angiosperms may be transported by bees or in the wind to the receptive female organs or stigmas of the flower. In mosses, transference is effected by rainwater. The antheridium discharges its sperms into the water film as a viscid mass. It breaks up into minute coiled sperms. Each swims by means of two cilia. The sperm swims in the water film till it reaches the archegonial shoot. The neck canal cells break down and form mucilage containing cane sugar, which attracts the sperms into the neck canal. One makes its way down the canal. And fuses with the ovum. The new sporophyte generation begins. Cleavage follows, and the embryo develops within the archegonial walls. At first, the embryo is a mass of cells. It soon shows three regions: a terminal sporogonial region, the seta or stalk, and the hostorium or foot, by which the young sporophyte obtains food from the gametophyte on which it is parasitic. The growing embryo ruptures the walls of the archegonium. The seta elongates to carry up the capsule. As it grows, it twists. The remains of the archegonium are carried up as a little cap, or calyptra. The capsules of various mosses differ in shape. But each capsule has its calyptra. It drops off later. Underneath, the capsule is covered by a lid, or operculum. This has a rim of cells called an annulus, and the lid breaks away around this rim. Here is another example of dehiscence. The spores are prevented from escaping even now by a dome-like arrangement of peristome teeth under the operculum. The teeth are hygroscopic. In damp weather they bend inwards and so close the opening. In dry weather they shrink and separate, and so the spores can escape. They are shaken out as the seta twists. Hypnum, another moss species, has a capsule rather different in shape and 2 layers of peristome teeth which separate completely as there is no central disc. The spores are very small, dry bodies. Their production marks the end of the sporophyte generation. The spores germinate in suitably damp surroundings into a new gametophyte. Each spore has a tough outer wall, or extine, and a thin inner wall, or intine. The intine pushes out through the extine, and the protoplasm moves into it as it grows forward. This is the first rhizoid. By rhizoids, the moss absorbs its food material. From the opposite side, another outgrowth containing chloroplasts develops, the protonema. From this green filament, buds arise to form the aerial parts of the new gametophyte. After about 24 hours, cross walls divide the thread into cells. Buds develop with their rhizoids at intervals and grow into new shoots. The protonemal connections will presently break down. Thus, from one spore, many shoots can arise close together. Hence the tuft-like habit of moss plants. Archegonia and Antheridia will be born at the tips of some of these, and so the cycle continues. The moss plant, which is the gametophyte, Gametophyte, gives rise to the male and female cells, sperms and ova, which fuse to form a zygote, Zygote. This develops into the sporogonium, Sporogonium, which produces spores and represents the sporophyte generation. These spores germinate to form a protonema. This produces buds which become gametophyte plants again with their own rhizoids and the protonemal connections between the buds breaking down. the closeness of the buds giving dense clumps of plants, facilitating sexual fusion.